Hello friends, welcome to Agriculture channel. I am Varsha Gaitonde and this video is about CRISPR-Cas technology, its applications. CRISPR-Cas is clustered regulatory interspersed short palindromic repeats. And along with this, Cas9 is also there. Here the two things are important. One is guide RNA and Cas9 protein. Guide RNA is an RNA which guides the protein to go and bind at a specific site. The two processes are involved here. One is homologous recombination and non-homologous end joining. In non-homologous end joining, the mechanism is by natural repair system. A particular defect is created or a particular defect is overcome by just a simple mutation and natural repair process, whereas in homologous recombination, by cutting a specific sequence, introducing a DNA of our interest will give the recovery of that particular site. This is a major approach of gene therapy. Cas9 just acts as an endonuclease which goes and gives a cut. That is all about CRISPR. Let us see how it works. CRISPR is an emerging game-changing molecular tinkering tool, certainly a major technique for rapid and targeted strategies. Development of CRISPR complex is just an art and science of prolific understanding and is an over one-time investment. This is well known for its simplicity, efficiency, minimal off-target effects and multiplexing nature. It was being used for gene knockout, replacement, genome editing, controlling gene expression, interrogating gene function and transcription modulation. In agriculture, it may help to develop location-specific and climate-resilient crops and enhancing the crop quality and quantity. However, there is a need of regulations. CRISPR stands for one of the discovery during the whole genome sequencing in 1980s when researchers of Osaka University sequenced the genomes of common bacteria. They found that repeating DNA sequence in many species but did not know its biological purpose. But in 2013, 3 different research groups simultaneously reported the use of this system for targeted genomic modifications in plants. Later, a number of other reports demonstrated that efficacy or potential of CRISPR-Cas9 technology as an efficient genome editing system in model crop plants. Majority of the CRISPR techniques were experienced on Arabidopsis and tobacco plant, but now it has extended to different crops also since 2015. Since 2014, huge research is promoted to improve in agriculture sector. Types of CRISPR. The system is categorized into 3 distinct types, that is type 2, type 3. On the basis of the presence of specific signal Cas protein, it is differentiated. All the 3 types have Cas1 and Cas2 proteins in common. However, the effector complex that binds to CRRNA and triggers cleavage differs among different CRISPR systems. Type 1 system is found both in bacteria and archaea and targets DNA sequences with the help of endonuclease activity of Cas3 protein. Type 2 CRISPR-Cas system has been reported only in bacteria. This you need to remember that type 1 system is found in bacteria and archaea, whereas type 2 is found in only in bacteria. It involves several proteins like Cas1, Cas2, Cas9, Cas4, CSN2, among which Cas9 is most efficient, significant and multifunctional protein. Type 3 system, which is recognized by the presence of Cas10 and Cas6 proteins in addition to repeat associated mysterious protein of RAMPs. Coming to the application part, the first and foremost is gene therapy. For every gene therapy, CRISPR is a medicine or it is utilized efficiently. In gene silencing, The approach has also been adopted for preventing the interaction between key cis-acting DNA motifs and their corresponding trans-acting transcription factors in the promoter regions to block transcription initiation. The gene knockdown by CRISPR is highly specific and reversible in action. Second application is gene knockouts. Knockout means just get rid of. It can eliminate genes that affect food quality, divert energy away from valuable end products, and confer susceptibility to crop diseases. For example, if a particular susceptible gene is present, or if a particular gene is responsible for the rice spoiling, that means by eliminating that particular gene sequence where that gene is residing, that is nothing but gene knockout, so that we can get a quality food. Using Cas9 enzyme, powerful ability to enhance or suppress gene activity could touch on many important processes of crop and livestock metabolism, resistance, and yield, generating conditional alleles, providing spatial and temporal control over gene expression to study the function of lethal genes. It also gives a गाइडलाइंस मिलता है कि हाउ लीथल जीन्स एक्ट ऑन एन इंडिविजुअल, तो उसको भी गेट रीड कर सकते हैं. Editing via recruitment of proteins responsible for histone modification or DNA methylation, which could open an entirely new avenue of regulation of cellular function in plants. अगर ओवरऑल बताया जाए तो ये एक जीनोम के ऊपर कंट्रोल पा सकता है, because when histone modification and DNA methylation kind of processes are controlled, तो पूरे जीनोम के ऊपर काबू पाना कोई बड़ी बात नहीं है. Similarly, this approach could be established as a useful tool to identify regulatory proteins that bind to specific DNA sequence to control gene expression. Next application है stress resistance में. Stress has a complex environmental interaction. Gene function depends on precipitation, heat, nature, depth of soil, and genetic background. CRISPR has an ability to address few tasks at a time. मतलब मल्टीप्लेक्सिंग का मतलब ये होता है बहुत सारे टास्क्स को एक ही साथ एड्रेस करना. The system promotes marker genes and antibiotic selection free genome engineering with high precision in diverse plant species used in developing viral induced gene silencing, example in case of tomato. It confers enhanced resistance to the plant against Gemini virus species. CRISPR system does not require any protein engineering and rarely cloning steps like the other editing techniques. Researchers can engineer large genomic deletions or inversions by targeting two widely spaced cleavage sites on the same chromosome. मतलब एक टाइम में टैलेन मेगा न्यूक्लियसेस और ZFN में क्या होता है हियर, और अगर आप जेनेटिक इंजीनियरिंग की बात करें तो एक ट्रेट या एक पर्टिकुलर सीक्वेंस को एक ही बार टारगेट किया जाता है. बट इन केस ऑफ क्रिस्पर ये मल्टीप्लेक्सिंग एबिलिटी रखता है और लार्ज जीनोमिक डिलीशंस इन्वर्शन को भी सेम क्रोमोसोम में टू डिफरेंट पोजीशन में हम टारगेट कर सकते हैं. The particular genes used will only work well in certain genetic backgrounds and environments. If we want to design agriculture for local ecosystems suited to the specific soils, climates, and cultivation practices of local people, CRISPR is at best partial solution, like changing it into genome site specific cultivars or repressors of gene transcription. ट्रांसजेनिक्स में ये यूटिलाइज किया जाता है, जैसे कि वायरल डिलीवरी ऑफ द कंस्ट्रक्ट्स और क्रॉसिंग ऑफ द ट्रांसजेनिक प्लांट्स carrying the CRISPR-Cas system with allied line cultivars can provide non-transgenic genome modifications in plants, which can address the ethical concerns being raised by transgenic plants. It is possible to segregate and remove the transgenes used during the editing process so that subsequent generations of plants are indistinguishable in their makeup from plants which have been conventionally bred. The CRISPR editing methods can be used to delete tDNAs for non-transgenic sweet potato generations to test whether the tDNA confer useful phenotype in naturally transgenic sweet potato. Inducing crop diversity. In this application, CRISPR can be used to create a high degree of genetic variability at a precise locus in the genome of the crop plants. It is a potential tool for multiplexed reverse and forward genetic studies. Next application is fluorescent tagging. An inactive Cas9 fused with an enhanced green fluorescent protein was co-expressed with specific single guide RNA to enrich the fluorescent signal at the target genomic loci, which demonstrated the use of CRISPR system as a robust and flexible platform for visualization of genetic element dynamics. It helps in identifying the chromosomal modifications in the entire life cycle of the plant under experiments, not only in plants, in animal genetics also it is helpful. It also explores environmental stimuli, sorting of specific cell types by tagging them with a fluorescent marker to facilitate the study of various functional aspects. It has been exploited to purify a specific portion of the chromatin and identify the associated proteins, thus elucidating their regulatory rules in transcription. Next application is in nitrogen fixation. Here, targeted mutagenesis used by CRISPR-Cas system can bring advances in functional genomic research of legumes, especially by generating the target mutants of the genes involved in roots and nodules, enhances the efficiency of nitrogen fixation. To reduce acidity in apple, then to inducing the polyploidy used in many crops across the world. This technology can also be harnessed to produce alteration in the histone protein found in centromeric nucleosomes, which can lead to selective loss of one set of chromosomes, or chromosomal elimination technique we can say. This approach can be utilized for the production of haploids, through which polyploid generation will be easy. Example, in case of potato and wheat. CRISPR technology has been used to mutate selectively only one of the three homoalleles of mildew-resistant locus, that is MLO-A1, in hexaploid wheat with high mutation frequency. In addition to genome editing, CRISPR-Cas system has been employed to regulate gene expression. There is a further scope to multiply the pharmaceutical drugs and mass production of it into general cultivable plants, which may drastically reduce the cost of several drugs used to treat human and animal diseases, as well as the drugs for treating the disease or for controlling the disease and pest control. The primary problem associated with CRISPR is the probe alteration and off targets are certainly addressed recently by using alternative and combination of different Cas protein and RNA targets. Now one question arises, is CRISPR an alternative to plant breeding? Because our traditional crop breeding takes so many years in order to get a new variety which is adapted to a wider area and giving a stable performance over the years and over the locations. But CRISPR is not an alternative. Remember, conventional breeding technique permits breeders to meet their breeding goals with the aid of the tools like CRISPR, but conventional breeding remains necessary in order to integrate the traits into elite breeding lands. CRISPR should be understood not as a best approach to addressing sustainability chain challenges, but as a suitable tool that capitalizes on the knowledge that biologists gain through our ongoing study of nature. The two giant seed companies Monsanto and DuPont Pioneer has licensed CRISPR for generating the healthy seeds, but they are prohibited for gene drive, that is continuous inheritance of the transgene for a specific trait, and terminator technology, that is sterility in the progeny generation, makes farmer helpless to buy the fresh seeds every year, or farmer cannot multiply the seeds of their own. Thank you. I hope you understood this concept. If you want further more examples, it is already there is one more video called genome editing in my channel. You can visit that. Please do not hesitate or do not forget to like, comment, share and subscribe my channel.